Thank you very much for the message. Okay, so in theory now you should be able to see the screen. And in this seminar, I'm going to start introducing the world of mass spectrometry-driven spatial omics, in particular in the context of molecular pathology. So I think it's a good moment to pick up from effectively where Professor Fragetta left us the last time. So effectively introducing this concept of pathology 4.0. So now we think in modern pathology, there's already this influx, this infiltration of digital pathology techniques. This has already been combined with machine learning algorithms to recognize patterns in these histological images. But now, as we know, as we move into this era of more personalized medicine, this has been accompanied by molecular approaches. So already we know for a number of years, immunohistochemistry has had unfounded benefits in the field of pathology, and now we're moving towards quantitative immunohistochemistry. Also at the genomics level and the transcriptomics level, these are starting to be introduced to aid with the decision-making process. And one thing that we noticed that with a lot of these type of technologies is that they are spatial in nature. So they provide spatial context, which is also very important in the context of disease. So along these lines, there are also emerging omics techniques, such as mass spectrometry imaging, that have the potential to also support in this particular type of area. And I did a little bit of reflection myself and thought, okay, so why in the last 5-6 years or so has mass spectrometry imaging started to be associated with this field of molecular pathology? Because in all honesty, 10 years ago, there was a lot of hype with this technology. And you will see it been applied in a variety of different disease contexts. And then there was a lull, a certain stalling or progression of this technology. And this because there are a number of obstacles that needed to be overcome in order to render it more appropriate for the field or application in pathology. And one particular area that's starting to be addressed is the robustness of our methodologies. So already there are a number of studies which have evaluated in multicentric studies how repeatable, how reproducible are these methodologies, and highlighting that samples prepared in different centers with different instruments can produce reliable and reproducible classification results. And this can also be improved with the help of bioinformatical processing of that data. So by using algorithms which help to normalize data which has been generated at various different time points, we can further improve the robustness of our methodology. And I think also a prime example of this line of work comes from our group, our university in itself, in particular looking at thyroid cytopathology. And again, this work started a good number of years ago with these, let's say, famous feasibility or pilot studies, if you will. But then from there, from this collaboration between the mass spec individuals, the pathologists, the bioinformaticians, we started to try and address these areas. First thing, how can we improve sample quality to remove contaminants which may mask some of the more relevant, more important biomolecules within this type of samples? How can we improve throughput? How can we improve stability of these samples so they can then be shipped from various different centers, which we know in a clinical diagnostic context is very, very important. And then when we have these robust samples, this then means that we have the most adequate samples for the bioinformatic analysis. And then from there, we can generate these classifiers, which can be used to detect malignant or benign samples from various centers. Then another area where we've seen improvements is in spatial resolution. So of course, now we know this trend toward single cell omics is exploding again. And mass spectrometry imaging has also followed that lines. And now we have instrumentation in commercially available instrumentation where we can detect single cells based upon the molecular profile. And in some instances, also start to go into the subcellular level. And not only this, but acquisition rates. So the speed in which we can perform this analysis is increased, because it's great to have molecular information from a single cell. But of course, in clinical diagnostic workflow, it needs to all be done in a reliable time frame. So we've gone from the space of maybe days of analysis or even weeks of analysis 10 years ago to now the space of minutes, 35, 40 minutes. And then associated with this desire to reach the single cell level, there's also been the need for sensitivity. So now there's also this approach called MALDI-2, which is another form of post-ionization. So unfortunately, this type of ionization process isn't particularly efficient. So only a small amount of the molecules that are generated are actually charged. Many of them are neutral. So now in this cloud, you have a second laser, which fires and then ionizes a much greater amount of these molecules. And it's been shown to improve the sensibility of lipids and glycans, proteins, and among others. And then this also leads us to biocomputational approaches, which even in terms of UMAP-based algorithms or TS&E-based algorithms, there's been a lot of work which has been applied to mass spectrometry imaging data. But at least in my opinion, from what I've seen, the biggest promise lies with the help of machine learning algorithms. So there's been an excellent number of work that have performed unsupervised machine learning to detect the most relevant molecular features from within a complex pathological background. And again, I think a wonderful example, again, coming from our university itself, where they've used self-organizing maps to identify molecular cells of interest based upon their profile. I think in this image here also represents a wonderful example of how it could possibly be integrated with a pathologist's arsenal in the future. So here we have the output of these self-organizing maps, and then it could also lead the pathologists to highlight particular cells, which can then also be evaluated in more detail at the histopathological level. This is how we really need to see how this type of technology can be integrated in the future, a supportive tool. So after this pretext, now I want to really go back and explain briefly what mass spectrometry imaging is. Then I'd like to really go through some of the recent developments, effectively in the last year or so, which highlight the various different manners in which we can use this technology in the field of pathology and clinical research. And then finally present how we've applied this technology over the last 7 or 8 years, in particular in renal diseases. So let's start with mass spectrometry imaging. So again, this is the only slide that I have an overlap from 2 weeks ago. So effectively, mass spectrometry imaging gives us the potential to visualize the distribution of biological molecules in pathological tissue. So if you like, we can consider it as a form of highly specific form of molecular microscopy. And we can do this to try and unravel much of this complexity that we find in diseased tissue. And this is because we generate data in 3 dimensions, which can be represented as this data cube here. So these first 2 dimensions are our spatial coordinates. So effectively, the tissue gets rastered or segmented into these small pixels at the level of a single cell. And from which each of these pixels, we generate a mass spectrum or a molecular profile, such as the one that we see here. And in this mass spectrum, it represents the presence and the relative abundance of a biological molecule. So in one highly multiplexed analysis, we can generate upwards of 500 molecules, which means we have 500 tissue distributions or 500 different pieces to the jigsaw that we need to construct what's happening under that pathology so that we can try and get to the root cause. And in the scale of human disease, where is mass spectrometry imaging? On the whole, more or less, we are at the level of a single cell. So the limitations right now are between about 5 to 10 microns. But of course, we're characterizing our cells based upon the subcellular contents, the molecules that are inside, which are highly complex. And we know that the molecules which are present within these cells drive their function. And then the function of these cells effectively is what drives the destiny of the tissue. And as we start to try and unravel all of this complexity, it's becoming more and more evident that within all of this here lies crucial information that we can use to try and improve patient care due to personalized strategies. And in English, we say there's more than one way to skin a cat, which effectively means that, as the same with mass spectrometry imaging, there are multiple different types of it. There are multiple different technologies that we can use for various different approaches. And the most popular 3 are represented here. So DESI, which stands for desorption, electrospray ionization, and then we have SIMS, secondary ionization mass spectrometry, where you have a primary beam of ions, which is target on the surface of the sample, which ejects all these small elemental molecules. But from within them, we have MALDI. And effectively, in the field of pathology, it's most widely used due to its flexibility and capacity to detect a wide range of different biological molecules with a good level of spatial resolution and with an appropriate level of sensitivity. So from all of these mass spec imaging techniques, again, why MALDI in the field of pathology? And I think this is where I could possibly put myself at the mercy of any pathologists or clinicians who are in the room right now, because I start to try and link what are the characteristics of these type of instrumentation with the possible needs in the field of pathology. And we can link these to what are known as the 4S criteria, so these characteristics of the instrumentation. So quite commonly in certain pathological instances, it can require a multi-step process to be able to obtain a diagnosis. Histopathology, light microscopy, electron microscopy, immunofluorescence, immunohistochemistry. So in order for this technology to have a benefit for a pathologist, we need to do something that improves upon that. So we need to have something that's quick. And the new technology can provide this particular aspect. Then from time to time, we might have some instances where we have some subjectivity to the outcome of histology or immunohistochemistry. So we also need a technology that can remove some of this subjectivity. So if we increase the sensitivity or the dynamic range of the biomolecules that we're seeing, it can allow for more objective interpretation. Then at the same time, there may not always be sufficient information to obtain precise diagnostic, prognostic, or predictive information from that tissue. So we need something that's highly specific. And with modern mass spec imaging technology, we have this resolving power, so the ability to distinguish between very, very similar biological molecules. And again, of course, we have the need to detect molecular alterations in only small numbers of cells, or clusters, if you will. And this can be particularly important in cytopathology, for example. So what type of information are we effectively getting with mass spectrometry imaging? So one thing that I can specify as well, that why this technology has become so popular, is that it can be applied to a wide range of different samples. So both fresh frozen tissue samples, cytological samples, and now as well as formalin fixed paraffin embedded samples. And now with the help of more complex tissue preparation processes, which take advantage of on-tissue chemistry that are becoming more and more reproducible, is becoming more appropriate in multicentric studies. And then, of course, we perform our MALDI image analysis, through which we generate these beautiful, highly colored images. And from these images, we're deriving effectively two key pieces of information. So where are our biological molecules? And we can actually overlay our molecular information with histological image post-analysis, because the technique isn't destructive. So we can overlay the two pieces of information together. So we know where the molecule is and from which particular cell type it's derived. So in the example here, which we take from one of our studies focused on a medullary thyroid carcinoma, we highlight the distribution of thyroglobulin, which was trapped within these follicles inside the tumor nodule. Then, of course, we also have an idea of how much of that biological molecule there is. And I think what the beauty of this technique is that we don't have to consider just one molecule at a time. We can consider their accumulation of them. So the overall pattern of these hundreds of different molecules. So we strain away from this singular molecular identifier. And again, I think sometimes what's also forgotten a little bit about this technique is that it can also provide some important insights into how a disease is developing. Because with MALDI imaging, you get a molecular snapshot of a disease at any given time. And it may not necessarily be that the abundance of a biological molecule has changed, but its cellular localization has changed. And this example we take from one of the works that we generate in collaboration with Professor Limperio, where we studied patients with idiopathic membranous nephropathy who had an invaried response status. So in patients that responded, of course, we had the production of MIF, macrophage migration inhibitory factor, within the inflammatory regions of the tissue. But then in patients that didn't respond to the treatment, we didn't see a huge overexpression, but simply an accumulation in and around the glomeruli. And then when we tried to validate this with immunohistochemistry, we found positivity at the podocytes and the parietal epithelial cells. But because we also knew the name of the protein, we knew what the protein was, we could then start to think of a molecular hypothesis to how this was deriving its function. So we hypothesized that MIF was able to bind to the CD74 receptor, which quite commonly is expressed on the surface of podocytes and parietal epithelial cells in cases of nephropathy, to stimulate this PLA2 pathway, which in doing so overrides some of this NF-kappa beta pathway, which is usually mediated by glucocorticoids. So after this brief introduction to what the technology is, now I want to focus on maybe some of the more recent advancements. So all of the work is effectively from the last year or so. And just to highlight the various different manners in which you can use this technology in the field of pathology and for disease understanding. So when I think of the possible uses of this technology, I think of Janus Bifron, so the Roman deity who has two faces. That's because I think there are two users effectively of this type of technology. So the first is tissue typing. So use this approach to try and detect molecularly distinct regions of tissue that we can then correlate with clinical endpoints. But at the same time, we can also use it in a slightly different way for molecular understanding. There is a little bit of an overlap between the two, but we'll see the effective difference between them afterwards. So focusing on this area of tissue typing, again, from here, we can use the technology in two different ways, in an untargeted manner. So I already presented some of our work about 2 weeks ago, focused on the lipidome, also the N-glycone. So we know the type of sugar molecules which are expressed on the surface of proteins which can affect their solubility and therefore their quaternary state and therefore their function, which then has an impact on how well the cell is interacting with others, cell-cell adhesion and so forth. And this type of work is becoming increasingly promising in cancer diagnostics, in particular those glycose structures which have unbranched manostructures high up within their chain. But of course, we can also use this for spatial proteomics. But not only this, we can now use it in a more targeted way to specifically search for known cell surface expression markers in a multiplex manner. But let's start with this untargeted spatial omics. And this is a line of work that we developed last year, which involved the use of a sequential workflow to perform spatial multiomics on a single tissue section. Because it's become more and more evident that various different biomolecules contribute to a disease in different ways. And when we can combine classifiers which span multiple omics levels, we can improve the stratification of patients. And again, this is particularly important that we're able to do this on a single tissue section, because from time to time, with certain rare diseases, diagnostic material is quite scant. In an instance where we might have heterogeneous populations of lymphocytes within that tissue, there are differences in sequential tissue sections. So we set up this workflow to be able to do it all on a single tissue section. The first thing that we did is we wanted to assess its feasibility. So in order to do this, we started with a standard tissue, which is some murine brain tissue that we obtained in collaboration with a group of Professor Cavalletti. And here, these are just three examples from three technical replicates. And we find straight away that the different biological molecules tended to be able to highlight the various different regions of the brain differently. So for example, we had the lipids and the tryptic peptides, which on the whole tended to better underline hippocampal formation, whereas these N-glycans better underline some of the structures within the cortex. And for example, from among this, we had the myelin basic protein, which highlighted the white matter, for example, and various histones, which highlighted the nuclei-rich region of the gray matter in the brain. And again, of course, we evaluated the reproducibility of this method as well. And we found that on the whole, the average coefficient of variance of any of these molecular peaks was below 20%, which is in line with what's been reported with this technology at a single omics level. So of course, then we applied this to a more complex type of tissue. So quite commonly in our lab, our research focused on clear cell renal cell carcinoma. So we use this type of tissue as a proof of context. And again, the first thing that stuck out to straight away is that the different biological molecules underline different regions of tissue. So for example, the lipids tended to distinguish the tumor-infiltrating lymphocytes and the leukocytes from the rest of the tissue quite nicely. whereas the N-glycans, as probably expected, enabled us to separate the tumor tissue from the non-tumor tissue, whereas the proteins, or the tryptic peptides derived from these proteins, the capsule, so there's accumulation of connective tissue, and the inflammatory regions quite well. And in addition to this, the workflow, as you saw, had multiple different steps, multiple different washing steps, and despite this, the molecules stayed in place. So for example, here we have the mentin, which we know is a glomerular marker, and we find it remains quite nicely localized, these glomeruli within the normal region of the cortex. And then myosin, for example, a protein who we know's expression may be correlated with the presence of tumor-infiltrating lymphocytes, remain very nicely localized to this particular region in the tissue. But the real goal is, what is the advantage of integrating all of this information together? So this is what we started to then try to address. So we took this complex region or complex section of CCRCC tissue section, and then we performed principal component analysis considering each of the omics levels individually. And again, as mentioned, some of them separate better the tumor-infiltrated lymphocytes from the rest of the tissue, some of them separate the tumor from the normal tissue, and so forth. But then what happens when we then take all of these features and combine them together and try and reperform this principal component analysis? As we see, when we combine this together in a multiomics data set, it was the only instance where we were able to successfully distinguish all of these regions in the tissue based upon their molecular profile. And again, to further highlight the advantage of this multiomics approach, here we have the loadings plot of the top 10 most contributing features from the single omics data set and then the multiomics data set. So when we confront the multiomics data set, we have a phosphatidylanocetal, then we have a protein, then we have another protein, then we also have some hexnap structures, so N-glycan structures here. So they were all contributing. There wasn't one molecule that outperformed the others. They all were contributing to the separation. And we hope then in the future, because this was only established last year, that we can then start to apply this in oncological context, in particular diseases where there's quite a complex immune environment that may then be correlated with chemo resistance. Then another particular novelty, which has come out in the area recently, is the use of these photocleavable mass tags, which have been attached to antibodies. And these can actually be dual-labeled antibodies, which means that you can apply a mass tag and a fluorophore to the same antibody. And you can analyze up to 25 tagged antibodies within a single analysis. So there's no need for multiple stripping, but all within a single analysis. And one of the advantages of this approach as well is that it can be compatible with the untargeted spatial omics analysis that I've shown previously. So first, you could analyze the lipid heterogeneity within the tissue and then see how this correlates to these specific cell surface expression markers. So again, this is an example that we generated recently as well in collaboration with the company that is producing these antibodies. And again, this is just an example of a small number of these tagged antibodies in a breast cancer data set. But again, we know that HER2, it's also important to see the subcellular distribution of this from a diagnostic concept. So where it lies within the cytoplasm. So this you could tag with a fluorophore, look it under immunofluorescence, and then afterwards perform this mass spec imaging using these mass tag labels. And this was also taken forward very, very recently by then to try and use the distribution of these antibodies to guide more in-depth, detailed mass spectrometry analysis. So a panel of a number of different antibodies were applied to the breast cancer tissue, and then a clustering algorithm was used to try and determine which were the phenotypically similar cells. Then from this clustering algorithm, they were able to dissect the cells, cut them away, and then use LCM-SMS-based proteomics to generate a list of hundreds and thousands of proteins, which were then interrogated with network analysis. Again, so this is another example, but is maybe more so from a clinical research standing. But the reason why I mention this is because it also then leads us to this secondary use of mass spectrometry imaging for molecular understanding. Because I would be amiss, of course, standing here saying that mass spectrometry imaging is the perfect technology, you can do everything, it doesn't have limitations. Because yes, you obtain this spatial context, which is very, very important. if you want to try and identify all of the molecules which may be present, it's not the most appropriate technique. So we can use it in a second way. We can use this as a molecular guide. So, for example, we can perform that untargeted spatial proteomics, for example, to highlight phenotypically distinct regions within a complex oncological background. And then from this, we can dissect these pure cell subpopulations and perform more in-depth omics characterization using complementary MS-based techniques. So we could then interrogate the lipidome, the metabolome, and the glycome, for example. And this is highlighted within a recent paper by Duallabeled et al., where they first performed immunohistochemistry on a breast cancer tissue section with Ki-67. And from this, they found effectively a relatively homogeneous tumor cell population. But then when they performed mass spectrometry imaging, they actually highlighted heterogeneity within this region. Of course, they then wanted to know, okay, why was the heterogeneity? So they then dissected all of these cells corresponding with each subpopulation and performed more in-detail proteomics analysis. So in each of these cell clusters, they generated a large number of proteins that were specific for that type of cell. All type of information that would have been lost if you used immunohistochemistry as a guide, or histology a guide, or perform bulk omics analysis where you mixed all of this tissue together and lost some of this spatial context. And again, within our laboratory, this is something that we've also recently established, but at a lipidomics level. So rather than using proteins as a guide, we've used lipids as a guide to highlight regions of tissue which are appropriate for this laser capture microdissection. So first of all, we perform our spatial lipidomics imaging to highlight some various regions in the tissue. Here we've just generated some very general regions, such as the hemorrhaged region of the tumor nodule, the rest of the tumor nodule, and then this cortical region here. And then from each of these regions, we perform laser capture microdissection of approximately 25,000 cells. These have then been accumulated in an Eppendorf tube, and we've then used liquid chromatography coupled with tandem mass spectrometry to identify all of the lipids and small molecules that can be present. So again, what we've ended up with effectively are two separate data sets that have their own distinct characteristics and visualize slightly different classes of lipids. This is another advantage of this approach. You can increase the molecular coverage that you're able to obtain, and then you can combine them. So for example, all of those lipids that we've generated with MALDI, so in particular, we tend to see phosphatidyl inositol species when working in negative ion modalities, whereas with the LC-MS approach, we then detect large species of sphingomyelins and phosphatidylserine. So then what could we possibly do with this data afterwards? So how do we treat this type of data? So first of all, we can integrate them together, and we start from the spatial segmentation that we previously looked at. So effectively, all of these molecular profiles are grouped together based upon their similarity. And great, the first thing we can already see with histology. So we identify regions of the normal cortex, regions of fibrosis, regions of hemorrhaging. But then from within the histologically indistinct region of the tumor, we detect two different molecular regions, something that histology wasn't able to underline. And then from this, we can then elaborate this data further. So for example, performing principal component analysis to confirm, okay, there are differences in these overall molecular profiles. And then because we have the list of all of these metabolites and lipids together, we can then link this together with the spatial information. And again, this is an example of here as something where we've gone in with a specific clinical question in mind, but highlights what can be done with the technology and how we aim to employ it within the future within an oncological background. So then the final area that I'm going to look at is in the application of renal diseases. So again, this is the line of work which I focused primarily on in the last 8-9 years. In particular, I work quite frequently with Professor Limperio and Professor Pagni. And this all started in 2014 with the renal biopsy. So we know that the renal biopsy still remains a fundamental component of the diagnostic evaluation of renal diseases. And it provides us with significant amount of information regarding the pathological state of that tissue. However, it's not always capable of detecting changes which can explain prognostic factors or even predictive factors. So will a patient respond to a therapeutic treatment. So of course, as we know, it's quite often accompanied with molecular techniques. So for example, immunohistochemistry, in the instance of membranous nephropathy, if we have positivity for phospholipase A2 receptor, we know it's idiopathic rather than a secondary cause. Of course, then we know we use immunofluorescence and a variety of other molecular techniques. But what will happen if we take the same renal biopsy and we apply MALDI imaging technology? So in one single analysis, we will generate hundreds of proteins, each which correlate with a specific feature within that kidney. So we will detect tens and hundreds of proteins which co-localize to the glomeruli and the severity of lesions which might be present within there. All of the proteins which are associated with interstitial regions of sclerosis, for example, proteins which may be responsible for inflammation or a variety of other lesions which we may find within the tubular interstitial areas, which may also have a prognostic impact. And as mentioned previously, this work stemmed back to 2014, and we've been developing it ever since then. So back in 2014, we first applied this technology to fresh frozen tissue, and we demonstrated its feasibility. So within various forms of glomerulonephritis, so minimal change disease, IgA nephropathy, we say, okay, within the glomeruli, we have different protein profiles from the tubules, and these correlated with disease severity, for example. Then in 2016, we were fortunate enough to be part of a European network, so IMODE-CKD, which came under the umbrella of a Marie Curie project, where we were able to focus on this in a little bit more detail. And one of the works that we performed highlighted the increased expression of alpha-1 antitrypsin within sclerotic glomeruli of diseases. And then in collaboration with the Biomedical Research Foundation Academy of Athens, we then correlated this with the urinary proteomics or peptidomics profile of the patients. And we found an inverse correlation between the expression of this protein and the estimated glomerular filtration rate. And again, which is a nice example of highlighting how we can go from tissue-based proteomics to a less invasive test. And again, in 2017, work that was very, very important to us is that we first developed protocols that we could apply this technology to formalin fix paraffin-embedded tissue. And again, this meant that now we can be involved in multicentric studies and receive samples from all across Italy and Europe. And in fact, one of the disease contexts that we applied this to first was in membranous nephropathy, where we generated protein profiles of glomeruli to distinguish idiopathic cases from secondary cases. This then continued in 2018, where I guess we entered in this personalized medicine era or personalized medicine scope, if you will. Whereas mentioned, we then used this technology to try and find proteomic indicators which could predict response to immunosuppressive treatment in cases of membranous nephropathy. And then in 2019, in collaboration with Leiden University Medical Center and the University of Calgary, we then detected specific molecular profiles of patients with diabetic kidney disease and were able to distinguish them from hypertensive causes based upon the presence of the overexpression of progesterone receptor membrane component 1, which we believe to be associated with a hypoxic environment which can occur in this type of disease, as well as the overexpression of certain complement-related proteins, which has started to be indicated as a possible pathological mechanism of this disease. Then in 2019, we also started to want to delve deeper within the glomeruli and take advantage of this high spatial resolution capability. So in addition to being able to generate specific protein profiles of glomeruli entirely in tissue, also specific protein profiles of the cells from within inside the glomerulus, enabled to distinguish from mesangial cells and podocytes, for example. And then this leads us to 2022, where we now embarking upon another approach by using this technology, which has also helped with a recent grant from La Ricerca Finalizzata that we've been very fortunate to receive, where we're starting to look at, can we use this as an in situ approach for the detection of renal amyloidosis? So the last 5 to 10 minutes or so, I will just briefly present the results that we've recently obtained. So of course we know, as a general rule of thumb, renal amyloidosis is detected with the help of Congo red staining, which can be highlighted with the help of light microscopy or polarized light microscopy, which leads to the presence of this green birefringence. And it enables us to detect these misfolded proteins which have accumulated in the form of these plaques or deposits. And these occur as a result of a misfolding of a variety of different proteins, such as serum amyloid A, immunoglobulin like lambda, immunoglobulin like kappa, among others. And being able to distinguish the type of this precursor protein, of course, has some quite important diagnostic and prognostic relevance, which is why more frequently highly specific MS-based approaches have been used to be able to define the type of precursor protein that's present within these type of patients. But in addition to these precursor proteins, we also have some amyloid-associated proteins, such as various apolipoproteins, vitronectin, and serum amyloid P component, which also co-localize to these regions of renal amyloidosis. And for us, this was great because on the whole, MALDI imaging technology is quite adapted to detecting this family of proteins. So we thought, okay, can we use this technology using the presence of these proteins as a guide to detect regions of renal amyloidosis and define the responsible precursor protein that can then be integrated quite nicely within a pathologist toolbox. So the first thing that we did is we performed spatial segmentation of our tissue. So effectively, this means that we grouped all of the spectra together based upon their similarity, which means that all those regions where all of these fingerprint-associated proteins co-localized were highlighted in a certain way. And when we did this on an example renal biopsy here, we found that these regions in green here were quite readily highlighted, which we presume correlated with glomerular amyloid deposits. Of course, from there, we're then confronted with the renal pathologist, and he highlighted these amyloidotic glomeruli within the tissue, and there was quite a nice correspondence between the automatic segmentation based upon the MALDI imaging data and the Congo red staining. So the first step was, okay, this can be a feasible tool which can enable the automatic detection of these regions of renal amyloidosis. But then what about detecting the specific precursor proteins? So again, here we had cases of AA amyloidosis, AL kappa amyloidosis, and AL lambda amyloidosis. So in the case of AA amyloidosis, we had a clear increase in SAA, whereas in the form of AL kappa amyloidosis, we had a large increase in the IG kappa protein, whereas in the lambda form of the amyloidosis, we had quite a distinct increase in its precursor protein. All of these were highlighted with a p-value below 0.05. So, okay, it can also be an appropriate tool for detecting the specific precursor proteins in renal amyloidosis. But again, the response here was, great, you've said something the pathologist is already able to tell you. Where's the advantage? What's the benefit of this type of approach? So now we started to tackle some more challenging cases. So we provided with cases of minimal amyloid deposition, which meant there was only a very small amount within the tissue, which was representative of the early stage. And then we had cases with light chain deposition disease, which meant that we were going to have the presence of these immunoglobulin light chains within the tissue, which can sometimes lead to a diagnostic interference, but without the deposition of these fingerprint proteins. And then when we performed the imaging of these two types of tissue, we found in the case of the minimal deposition, we found an accumulation of these fingerprint proteins all within the same region, which are highlighted in black here. Whereas in the case of the light chain deposition disease, these were distributed diffusely within the tubular interstitium. Again, here, the glomeruli are highlighted in black, and we don't have them all co-localizing within the same area, which is what you would expect with this type of disease. And again, highlights where spatial context is important, because if we mix these two pieces, all of this tissue together, maybe this wouldn't have been highlighted. But then from here, okay, can we then also detect the correct precursor protein? So both of these cases of light chain deposition disease, we knew were cases of lambda light chain deposition disease. As we can see here, there was quite a distinct increase of the lambda protein as we expected to see. Then we had these two cases of minimal amyloidosis. So minimal amyloidosis, there was a suspected amount of the lambda form of the disease. And again, straight away from the MALDI imaging data, we saw a distinct increase in the lambda protein. And then we had this interesting case of MA1, where at the time of diagnosis, the responsible precursor protein couldn't be determined. It was only a number of years later, upon further clinical manifestation, that they were able to determine that it was actually the lambda protein that was responsible. And on the renal biopsy that was used for the diagnosis at the initial time point, we saw this increased expression of the lambda protein itself. So again, highlights where this increased sensitivity can be useful for renal pathologists in trying to support them in this process, especially when you have cases of amyloidosis that are only in the early stages of formation. And of course, this work is relatively early, but again, we're quite happy with the promising results that we're obtaining. But a lot of this work as well has been possible with the help of the nephropathy platform that we've recently set up. And so here, I think, again, it's a great example of multidisciplinary research. Because in this picture here, which I guess represents the heart of the platform, we have proteomics, those that work with proteomics, mass spectrometry. Then we also have those that work in the field of pathology. And then we also have those that work in the field of bioinformatics, among a whole host of other collaborations which are coming together in this project. And again, based upon my knowledge of what I see in the field of mass spectrometry imaging, this is quite a novelty, also on a global level as well. There aren't really these type of platforms that have been set up. So I think it gives us a really great opportunity to really try and push this technology closer towards the clinics. And I have 2 slides which remain. So what will happen if these 2 worlds of digital pathology and molecular pathology combine? Because in the previous seminar, we had Professor Fragetta highlighting the use of artificial intelligence algorithms to classify histological images. And in this paper here, so morphometric cell classification of single-cell MALDI imaging, this represents this quite nicely. Because they started with regions of tissue sections of porcine colon, and they had a pathologist highlight regions of glandular cells and the lamina propria cells from within there, which in the zoomed in region here, unfortunately you can't see them very well, they're highlighted. This was then used to train a machine learning algorithm, then automatically detect in the rest of the tissue, okay, where are these glandular cells and where are the lamina propria cells? Then we introduced the MALDI imaging data, so the single cell MALDI imaging data, and we co-register them together. So not only do we identify the individual cell types that are there, but we could also have the potential to then individuate their specific molecular phenotype, which we can then use to try and guide more specific and precise diagnosis. Again, this is just an example. Of course, in the future, we want to apply this in a real diagnostic context, being able to detect different types of cells and their malignant nature, for examples. But it shows what can happen and what can be possible if these 2 pieces of technology combine. So the penultimate slide that I have is a bit of a stupid question, if you will, but fortunately doesn't warrant such a stupid answer. So the question is, are we there yet? Of course, the answer is no. But if I look at the progress that's been made, we've made large progress in the last 5 or 6 years. There still are some open questions that remain in terms of large-scale validation of the methodology. In the future as well, how could we store large amounts of data that have not only been generated from molecular data, but also pathology data, which still need to be addressed. But in the last 5 years, a large amount of difference has been made. But I mean, again, as we say in English, the proof will be in the pudding. And that proof will be in the future, not next year, not in 2 years' time, but will the technology be adopted by anatomy and pathology? So here I have the photo of Dr. Christina Schwamborn, who's a pathologist who works at the University of Munich, who has adopted this technology. Again, I have Christina's face, but it could be the face of our own Professor Pagni. It could be Professor Limperio, hopefully. And again, she's of the opinion that the technology is fast, is becoming ever more reproducible. And in fact, with respect to routine pathological workflows, it's not that much more expensive, which of course is always a crucial aspect. And she hopes that it has the potential to revolutionize pathology as a whole. This is what I hope for as well, and this is what we're going to try and push towards within the near future. So in addition to this line of work, we have a number of ongoing collaborations, some of them internally within the University of Milano Bicocca, some at some academic institutions within Italy, but as well within industry. In particular, we're currently running a study in collaboration with Illy Cafe as well, so the weird and wonderful of MALDI imaging, if you will, where we've been using this technology to try and detect bioactive compounds within coffee beans, so the molecules which provide the coffee with its specific properties. And in particular, we focused on the detection of chlorogenic acids, for example, and hopefully in the next 6 to 7 months, hopefully a nice article regarding this will come out. In addition to a national level, at the international level, we also have a number of ongoing collaborations as well. So we've recently started collaborating with the Koch Institute at MIT when we're using our spatial lipidomics technology to try and detect differences in HIV positive patients. And again, I'd also like to thank the rest of the members of the European Society of Toxical Pathology, which I make of a mass spectrometry imaging subgroup, which again helps provide me with a concept of what we're able to do with the technology, but also what a pathologist actually needs, a real world perspective to our technology. So I'd like to finish by thanking all members of our spatial omics group. And again, I always find this a little bit embarrassing that at the bottom we have the hardworking students, but they're always the ones that are running backward and forward, collecting samples for us, doing the grunt work that we don't want to do. But again, in particular, big thanks to Professor Pagni, Professor Limperio, Professor Galamberti, Dottoressa Capitoli, and some of our collaborators at Bruker as well, and finally you for your attention. Non so, allora, cosa devo fare ora? Okay. If I stop sharing, will be easier.
Grazie, veramente interessante. Vi chiedo se ci sono domande. Sì.
Okay, Andrew, first of all, thanks for the nice talking, very interesting. And maybe I didn't understand completely the subject, but and I have a question about the fact if you can stain a slide for your protein of interest and then isolate only those cells that are stained by your marker and analyze them.
Yeah, that's exactly correct. So you could perform this multiplex immunohistochemistry. Up to 25 markers right now. The vendor that produces.
No, I'm just talking about a regular antibody like the ones we use for regular immunostaining.
Yeah, in theory, yes. This is in fact something we're currently doing right now as well for another approach where the tissue has been stained with the antibody, and then we've used this thing guide laser capture microdissection, then form proteomics on the isolated cells.
Okay. Thanks.
Also, thank you very much. It's a very fascinating technique, and it's a very interesting seminar. I have a curiosity more than a question. Have you experience? Please mute yourself. So my curiosity is, have you ever experienced this technique on a bone section? And if yes or not, I mean, if you're aware of anybody's doing this on the bone, because I'm thinking more of a less than a clinical, so not much as a clinical application, but as a basic molecular biology application. So if anybody has done it on the bone, and if not, or if yes, if there are any limitation related to the structure of the tissue. And that's my first question.
My answer is yes. We actually recently performed some work in collaboration with the University of Surrey, where they were interested in the proteomic distribution of certain fragments of collagen-related proteins in cartilage. Okay. So this is something that we've been able to do. As you can imagine, it's not the simplest tissue to work with,
yeah.
and it did require a certain degree of method optimization. It was actually mouse cartilage as well, so the size of the tissue was more or less minuscule. But in the end, we made it, and it seems that it is possible to work on this type of tissue. There are also some other groups as well that in the past have worked with fresh cartilage tissue. They've also performed lipidomics imaging on this tissue as well. So the answer is yes, it's possible.
Okay.
Problematic, not the easiest.
Yeah. Because I guess I'm also interested in doing this. So I probably should talk offline about that. But I know that the limitation is, first of all, the section, because it's very tiny, like especially on the mouse. And another question: have you ever done? Do you think it's possible to do tracing with the MALDI imaging? So doing like labeling carbons, if you're tracing, for instance, glucose pathways, let's say the incorporation of like how glucose is incorporated into different pathways, and you have labeled carbons. Are you able to detect these label fractions with MALDI? Like, I don't know, maybe I didn't explain myself correctly. So if you want to do like a glucose tracing that you usually do with the mass spec, do you think you can do it with MALDI as well? Just to do like the destiny of these carbons in
I'm
the tissue?
not sure specifically in terms of tracing, but I did see an article very, very recently that used an enzymatic approach to trace the presence of glucose within tissue. So I forget the enzyme that they used, but they used it to specifically liberate glucose or cleave glucose from the region of the cell, and then use mass spectrometry imaging to see where the glucose was present with
That's
the
also
tissue.
very interesting. Okay.
I think honestly, I forget the enzyme.
Okay, no, it's okay. It's okay.
This was also very. I think it was about 6 months ago that they
Okay, so very recent.
So probably it's a hot topic right now.
Yeah. Okay, thank you so much. Because that was interesting. I see. How many carbons do you have at the end? I see. Because I was interested in doing like fructose or glucose tracing and follow up on a serine synthesis, and serine is very small, and that only two carbons, I think. It will be excellent if we have like, where is it? Because I have, well, just to explain briefly, I would like to discriminate between, so I'm working with a model of leukemia that infiltrates the bone. So there is human in mouse, and there is normal and pathological tissue. So it will be very interesting for me understanding what kind of metabolites should be like, are produced by the tumors, what kind of metabolites from the normal tissue, and what kind of, because it depends also on where the leukemic cells home. So if they home in a specific part of the bone, for instance. So the spatial will be very interesting for me, just to understand also how the niche of this leukemia works. And then therefore, how do they metabolize glucose, fructose, or whatever sugar I'm studying. So that's why I was asking.
Okay.
But I know that's very, I guess it's very complicated to do, so.
Yeah, after me, the cartilage has probably been some of the more challenging tissue types that we've come. But I think it just requires a little bit of dedication. Everything performed on a micro scale.
Yeah.
Effective with this type of tissue.
Yeah. Thank you so much. Any other question? Yes.
Thank you so much for the interesting talk. I was wondering whether also applications to microarray readout are developing now fast by this kind of technique.
Yes,
there's a great potential also for, so not only for tissue, but also microarray.
Yeah, so in fact, a lot of our clinical studies are focused on the use of tissue microarrays. Of course, the amount of data that we generate is large, but it rapidly increases our throughput. So again, now if we want to perform these larger scale studies, we tend to ask for a tissue microarray. So again, effective, but the way we would treat the samples is the same way that we would treat the tissue.
Molecular microarray, not necessarily tissue, but for instance, the doubly labeled antibody with the fluorophore and the mass tag, would be a perfect tool also for my molecular microarray readout. Protein, for instance, protein microarray readout.
In theory, yes, the technology that's there would be perfect for this type of approach. From what I'm aware, nobody's done this so far. So again, if we're able to do that, that would be a particular novelty, I have to admit. Again, unfortunately, this dual-labeled antibody technology is something that's only really started to come to the front about a year ago. So there's only a small number of works which have started to develop, and this is something that we'd like to implement. We're trying to implement within our lab as well. Hopefully, within the next year, we'll be able to do that.
I have just a curiosity about the amyloidosis. You showed the about the renal disease.
Yeah.
You know, there is also this approach in other organ tissue, the amyloidosis.
Yeah. So it has been used in a variety of other tissues, so brain tissue, heart tissue as well. I think the first study was about 2015, where they showed its capacity to detect amyloid deposits in kidney, heart, and brain, if I recollect. And then quite commonly you see this correlated with amyloid plaques in neurodegenerative diseases quite frequently as well. And quite commonly they combine the lipidomics imaging with then the proteomics imaging, like where these plaques are, how they're constituted, and then how this has an impact upon the metabolomic profile of the surrounding cells as well.
Very interesting. Thank you. Other question? Thank you very much.
Posso chiudere ora? Perfetto.